All right, we are all ready to start. We are exactly on the hour. Good day, everybody, and thank you for joining our ZOOMinar series. And today we have two speakers, Dr. Sandrine Ongeri and Dr. Nicolo Tonali. We just please notice that there won't be a break for Q&A after Dr. Ongeri's presentation. I will just introduce Dr. Tonali after hers. And at the end, you can address both of them, and you can wait for once both speakers are done, you can wait to ask your questions. So it is my pleasure to present Dr. Sandrine Ongeri. She's a professor at the Paris Saclay University, CNRS. Dr. Ongeri did her PhD in the lab of Dr. Casson in the Department of Organic Chemistry at the University of Paris Descartes. During her PhD, she also spent 1 year at the Dyson Parents Laboratory in Oxford, UK, and she worked with Professor Sir Baldwin. After 2 years of postdoctoral research with Professor Gennari in Milan, Italy, Dr. Ongeri joined University Paris Saclay University. The main focus of her current research is the design, synthesis, and characterization of peptidomimetic inhibitors of amyloid protein aggregation with applications for neurodegenerative diseases and type 2 diabetes. And I don't want to steal any more of your time. It's your turn, and I'm so glad that you're presenting today.
Thank you. Thanks, Magda, for the introduction. Thank you, Magda and Rams, for the invitation. So today we will speak together with Nicolo about folded peptidomimetics based on cross interactions involved in the prevention of amyloid protein aggregation. So as you know, protein aggregation is involved in many degenerative diseases such as Alzheimer's disease, type 2 diabetes, Parkinson's disease, or Huntington's disease. And in our lab, we are particularly interested in 4 proteins, 2 proteins involved in Alzheimer's disease, A-beta and tau, IAP involved in type 2 diabetes, and alpha-synuclein in Parkinson's disease. So the main goal is to try to avoid the self-aggregation, but I will introduce some recent work about avoiding cross interactions or basing our design on cross interactions. So they have many common way of aggregating all these amyloid proteins, and we have been working on A-beta, IAP, and very recently in the frame of a consortium, a European consortium, to be trained, we have been working on alpha-synuclein and tau. The main objective is to try to avoid the presence of oligomers. As you know, as monomeric, amyloid proteins are not toxic, while when they aggregate, they form toxic oligomers and fibers, and we try to avoid the presence of these toxic oligomers by preserving the monomeric species. This should allow to increase the clearance of these pathogenic amyloid proteins. So for that purpose, we base our design on the conformational preferences of these amyloid proteins. They are intrinsically disordered, but they start to adopt some helical conformation when they are monomeric, and then they misfold to form beta sheet-rich structures, oligomers, and fibers. So the idea is to try to trap monomeric species or to avoid the presence of small oligomers in order to maintain the monomeric states. So for that purpose, we design mimics of helix to try to trap the monomeric species of amyloid proteins. And we have worked a lot on mimicking beta-hairpin compounds in order to destroy and to try to avoid intra and intermolecular interactions between these beta sheet amyloid structures. So the idea is to try to rationally design folded peptidomimetics to inhibit amyloid protein aggregation. For that purpose, we have specific interest in the monomeric, in the oligomers, and in the fibril structures we can find in the literature. So from NMR, cryo-EM, molecular modeling, we study the interaction and the specific peptide sequence involved in the beta sheet structures, and we try to mimic this peptide sequence in order to be specific for each amyloid protein. So for example, for A-beta here, you can see that we have a specific amyloidogenic sequences between Ka16 and F20. And we have a look also on the facing sequences in the beta sheet structures. So for example, here you have in front of KLVFF, you have this ILE to VAL36 residues. For IAP, we have this amyloidogenic sequence from asparagine 22 to leucine 27, and we have a look on different reported structures to try to see what is facing to this amyloidogenic sequence. And here you can see that we have facing alanine 13 to histidine 18. So to design beta-hairpin peptidomimetics, the idea is to introduce mimics of beta term, so unnatural beta term inducer, and we put some peptide sequence from the specific amyloidogenic sequences and the facing sequences. The idea is to try to pre-organize compounds, because as you know, when you use only small peptides, they are not able to inhibit the aggregation, or they are not very efficient to inhibit the aggregation. So by pre-organizing hairpins, the question was, can we target monomers and small oligomers and avoid the misfolding and then the aggregation? For A-beta 142, we have used two different beta term mimics. The first one was based on the diketopiperazine beta term mimics, and we bind to this beta term mimics some amyloidogenic sequence from A-beta and a disrupting beta, disrupting peptidomimetic strand. In that case, we had a very stable beta-hairpin mimics. However, it was not very good to inhibit A-beta aggregations, probably because it's too stable. As you know, we have different conformations. So the idea is to try to maybe use beta-hairpin mimics more flexible to try to adapt to the different beta sheet conformation of amyloid protein. So we change the beta term mimics, and we use this beta term mimics based on piperidine pyrrolidine ring. In that case, we have at least two different conformations where the peptides can adapt to the beta sheet structures of A-beta. So we put the amyloidogenic sequence, KLVFF, and in phase, we used the facing sequence found in oligomeric structures of A-beta. In that case, we had very, very good inhibitors of A-beta. We then decrease the peptidic character, and we obtain, again, good to very good inhibitors. This compound was selective for A-beta. We used peptide sequence from A-beta, and it was not active at all on IAP. Then, later, we used the same beta term mimics, but we introduced peptide sequences from IAP. So for the sequence recognition element, the amyloidogenic sequence of IAP is from N22 to L27, and facing, we use the sequence from phenylalanine 15 to S20. In that case, we were able to inhibit or to decrease the aggregation of IAP, but we were very selective and no activity at all on A-beta. So beta-hairpin mimics based on specific sequences of amyloidogenic proteins can inhibit selectively the aggregation of the proteins. We then decrease the peptide character by reducing the size of the peptide and by introducing other amino acids. More recently, we have tested different compounds that we call foldamers. So foldamers are mimics of peptides where we add 1, 2, 3 or more units, unnatural units. In that case, we use these difluoromethyltriazoles because we know that we have already shown that this scaffold is able to impulse, to push the peptide to adopt an extended conformation. In that case, when we add 3 to 4 units, we obtain extended compounds, but they increase the aggregation of IAP. While when we introduce between 2 units, we introduce some natural amino acids from the amyloidogenic sequence of IAP, and in that case, we obtain beta-hairpin structures that were able to decrease the aggregation of IAP. And they were selective, no activity at all on A-beta. Even more recently, we have decided to try to trap the monomeric conformation of amyloid proteins. So the idea was to try to mimic helical structures to interact with the helical conformation of the amyloid proteins. In that case, it was A-beta, but it worked also on IAP in order to avoid really the misfolding in beta-sheet-rich compounds. We took some compounds on foldamers from a colleague in Montpellier, Ludovic Maillard. He gave to us different foldamers from dimer to hexamers, and he introduced different amino acids. And the most active one was the foldamers bearing lysine and leucine residues. The activity was quite good to inhibit both IAP and A-beta. So how do we evaluate our compounds? We evaluate both on the fibrillization and on the oligomerization, because as I told you in the first slide, we want to avoid the presence of oligomers because they are toxic. So to test the compounds, we first use classical techniques, THT, thioflavin T fluorescence, and TEM experiments. In that case, I illustrate some biophysical evaluations on these helical mimics, and you can see that it can decrease A-beta aggregations. Here you have the amyloid curve of A-beta alone, the control, and in the presence of the compounds, you can decrease the aggregation and you have the dose-dependent effect. On IAP, only at 10 to 1, this compound was able to decrease the aggregation. So the question was, do helix mimics can inhibit the aggregation or decrease the aggregation and trap the monomers? Yes, we can inhibit or decrease the fibrillization, but what about oligomers? So for that purpose, we developed 2 different techniques. The first one is capillary electrophoresis experiments, where you can see only soluble species, and in particular, we follow the monomers of IAP and A-beta. In the case of the control of IAP, here you can see that from 0 to 5 hours, you have a decrease of the monomeric species, and after 10 hours, you cannot see anymore the monomer. While in the presence of these helical mimics, you can see that the monomer is preserved a little bit until 15. It's more evident for A-beta. Here you have the control kinetics of A beta. So monomer species are completely invisible after 10 hours, while when you add these helical mimics, you can maintain at least 70% of the monomer even after 20 hours. So in these experiments, we can see that our compounds, our helical mimics, are probably able to trap monomeric species. We then studied by MS experiments if this compound is able to decrease the presence of very small oligomers. So in IAP control, you can see some dimers to trimers to hexamers. And in the presence of these helical compounds, you decrease the ratio of hexamers. Dimers is still present, but you decrease the ratio from trimer to hexamers. When you increase the ratio of dimers, you can nearly avoid the presence of all these small oligomers. Furthermore, you can see complexes, so non-covalent complexes between IAP monomers and foldamers. You can see that one monomer can bind to 1, 2, or 3 molecules of these inhibitors. So the first strategy we followed was, can we design compounds mimicking helix and beta hairpin based on the specific sequences of A beta and IAP? So this works. Can we now extend this design to other amyloid proteins, alpha-synuclein and tau? I will show you a few results on tau in a few minutes. And this is in course in the frame of our European network TubeInTrain. But more recently, we were very interested in a second strategy. This second strategy is, can we design folded peptidomimetics based on protein cross interactions that prevent the aggregation? Nicolo will explain just after me the interaction and the design of peptidomimetics based on A beta and TTR interactions, and I will show you now our recent work and unpublished work for now, results between tau and HSP90. Why were you in? Where can you design compounds from chaperone proteins? Because it is well known now that chaperone proteins in healthy cells can inhibit the aggregation of aberrant proteins misfolding. So molecular chaperones can avoid the aggregation. And these molecular chaperones, when you have age-related decline, are unable then to efficiently perform this function. So if we base our inhibitors on these molecular chaperones, maybe we could avoid the aggregation. So in the case of tau, we have selected the heat shock protein HSP90 that binds tau. And it is known, it is reported that this HSP90 can avoid the aggregation, especially at the early step of the aggregation of tau. HSP90 binds tau in the microtubule binding repeat domains, but we have decided to design inhibitors from specific sequences of HSP90. So what is known is that between HSP90 and tau, you have some specific and key residues that are involved in the interaction. And we have selected key residues in 2 strands. As we want to design and to use our beta hairpin mimics, so we will use the same beta turn inducer as we used in the previous strategy, but in that case, we will use specific sequence from HSP90. So not anymore from tau. We will compare the 2 strategies, but in that case, we have selected 2 different strands, S S4 and S7 because they have key residues in interaction with tau. We compare, of course, this design with the previous design. It means we also prepared some beta-hairpin mimics based on specific sequences of tau, PHF6 and PHF6 stars that are found in beta sheet structures when tau is monomeric and when it's binding to microtubules. And the third compounds is based on the structure of tau fibers from cryo-EM. So we will insert some specific sequence from beta-1 and beta-8 that are found interacting in fibers. The results by THT is very promising. We are very happy to see that with these three compounds, we have inhibition of the aggregation, but the most active compound is DL171, which is based on the peptide sequences of HSP90. So you can see here the amyloid, the fluorescence of A beta alone, and in the presence of DL171 based on HSP90, you can see that we can decrease dramatically, even at low ratio 1:1, the aggregation. Two others compounds are also efficient at 5:1, but much less efficient at 1:1. So using peptide sequences based on chaperone protein HSP90 is more efficient than sequences based on tau structures. Then we evaluated this compound on A beta 142 because it was reported many years ago, 15 years ago, that HSP90 is also a chaperone protein for A beta. So we used the same compounds, DL171, and we were very happy to see that DL171 is also able to decrease dramatically the aggregation of A beta 142. Here you have A beta alone, and in the presence of DL171, you decrease the aggregation. We test the isolated sequence, so S4 alone, S7 alone. In that case, we have no activity or very small activity of the peptide sequence alone. So the beta-hairpin structures is very crucial for the activity. However, I don't show here the curve, but when we test this compound on IAP, we didn't have any activity. It's not reported that HSP90 is a chaperone of IAP, or it's not well known, but in our case, it is not active on IAP. It's active on tau and A beta 142. Then we have a co-supervised vision of Nicolo. Nicolo Bisi is one of my PhD, working in collaboration with the University of Osnabrück in the frame of our European networks. And Nicolo did some experiments, some fluorescence decay experiments that are used to evaluate the binding of tau on microtubule. And what you can see here is that with the pro-aggregative mutant of tau, you have a decrease. You have only around 85% of this pro-aggregative mutant that is bound. Why? When we do the same experiment in the presence of the inhibitor, DL171, you can rescue the binding of this mutant to tau. So DL171 restores the physiological interaction between tau and microtubules. Furthermore, modeling of the kinetics, this has been done also by Nicolo in co-supervision with Roland Brandt. You can see that you have an increase of current. So it means, and it shows that DL171 increase the concentration of tau that is valuable for the interaction with microtubules. We have no effect on decay off. So this means that the compound does not directly act on the microtubules. He also very recently performed the same experiment on the wild type. So the wild type binds to microtubules, and in the presence of the inhibitors, we have no activity. So that means that DL171 does not alter the dynamics of the wild type tau and is selective against a pro-aggregative tau construct. Nicolo checked also that the compound is not toxic for the cells, and he didn't see any toxicity by LDH assays, neither no significant changes in metabolic activity when he performed MTT assays. So in conclusion, these very recent results based on HSP, on the chaperone protein HSP19 interaction with tau is promising to our point of view, because we can inhibit both proteins involved in Alzheimer's disease. What is in course? We have sent the compounds to Markus Svettekers in the Göttingen Max Planck Institute to evaluate the binding of our inhibitors with tau by NMR experiments. We are performing stability towards proteolysis, and we have also performing BBB crossing. The next step will be to do the same with alpha-synuclein. This is in course. We have also promising results of inhibiting alpha-synuclein aggregation based on the specific sequence of alpha-synuclein, but also on the chaperone protein. I hope I will be able to show you these new results, and I hope to see you in Catania to present these new results. So now I will let Nicolo explain to you what we have done on TTR and A-beta aggregation.
Hi, Nicolo. While Nicolo is setting up his presentation, I'm going to take a few minutes to introduce him. It is my pleasure to introduce Dr. Nicolo Tonali. He obtained his PhD in 2016 under the supervision of Professor Ongeri at Paris-Saclay University. He made his first postdoc in Germany in the laboratory of Professor Seewald at Bielefeld University, and a second postdoc in CEA Saclay in the laboratory of Dr. Servant. Dr. Tonali was recruited as assistant professor in medicinal chemistry in 2019 in Paris-Saclay University, and he's a young researcher and an expert in protein-protein interactions, particularly in the design of compounds derived from peptides, adopting conformations mimicking secondary structures such as beta sheets and helices of proteins in solutions, and their conformational studies by NMR, CD, and molecular dynamics. I'm not gonna take more of your time, and sorry, breaking up a little bit of the floor of your talks. It's your turn, Dr. Tonali. Nice to have you in the series.
Thank you so much, Magda, for the presentation, for the introduction. Thank you, Rams, for the kind invitation. And it's a pleasure for me to present today this project concerning a beta 142 peptide interacting with the transthyretin. So when we consider the cross interaction, in the amyloid field, we generally think about the process by which the amyloid aggregates of one protein are able to directly nucleate the aggregation, the amyloid aggregation of another protein. And this generally could happen, for example, for proteins such as beta 142 and the tau protein, which belongs to the same disease, so the Alzheimer's disease, because both mutually negatively influence each other. Or it can happen even for protein that belongs to different amyloid diseases, such as beta 142 in Alzheimer's and type 2 and IAP, which is involved in type 2 diabetes. So this type of molecular associations between different amyloid disease is what we call a negative cross interaction. But I'm sorry, I don't. Oh, but together with this negative cross interaction, there is another type of interaction that we can call a positive cross interaction that can be considered. This is the case of proteins which are responsible of some amyloidosis, but before their misfolding and their aggregation process, they can act as a sort of chaperones protein by preserving the physiological role of, or by controlling the correct clearance of another potential amyloid protein. For example, in Alzheimer's disease, there are proteins such as transthyretin, ApoA1, and cystatin C that play an important role in maintaining the beta in its monomeric state, so preventing its amyloid aggregation process, and even in participating in the physiological transport and metabolism of beta 142. In this presentation, I will speak about the TTR and the beta positive cross interaction and how we can take inspiration from this particular protein-protein interaction in order to design a new amyloid inhibitors. So transthyretin is a 55 kilodalton homotetramer, which is present in the plasma and in the cerebrospinal fluid, and whose main function is to transport retinol and thyroxine. So TTR is able to play its physiological role in its tetramer structure, but when the tetramer structure is compromised, the equilibrium is in favor of the monomeric form, and the monomeric form is more prone to aggregate. So this is why TTR is considered an amyloid protein, because it's responsible of systemic amyloidosis disease. In the field of Alzheimer's disease, TTR was proved to be also the major beta binding protein in the CSF. And it has been proved that the interaction with the beta 142 is able to inhibit the amyloid formation. As we can see here by the theophlavine T fluorescence assay, we can see that TTR in a concentration-dependent manner is able to delay or inhibit the formation of fibrils by beta 142. But the neuroprotective effect of TTR is not only linked to its ability to prevent the aggregation and the amyloid deposition, but also because TTR has a role in maintaining the homeostasis of beta into the brain, thus reducing the toxicity of beta 142. So there are two sequences that we can consider the key sequences of the interaction between TTR and beta 142. One is what we call strand G, from residue 102 to residue 117, and the other one is the one that we can call EF helix, that is a small peptide from residue 74 to 83. Both sequences contain two important key residues to leucine, one in 82 for the F helix and one in 110 for the strand G. And these two key residues are very important for the TTR interaction and amyloid inhibition. The two small peptides have been synthesized and isolated by a research group in Australia. And in literature is known that only strand G is the key sequence that can inhibit the aggregation process of A beta and can have an interaction with the beta 142. Because in their experiment, they showed that the F helix didn't show any type of effect on the beta kinetics. So the author suggested that probably this lack of affinity and activity of the EF helix sequence could be associated to the lack of alpha helical structure of the isolated peptide. And so they made the hypothesis that probably the helical structure is very important for the interaction. More recently, thanks to our collaboration with the University of Pisa, Dr. Ciccone could demonstrate the high affinity of the TTR in the presence of a 128 fragment of A beta 142. By using biolayer interferometry, it has been observed a KD of 54 nanomolar in the presence of copper ion, which stabilized the interaction between the two proteins. The crystal structure of the TTR in the presence of copper ion and the beta 128 have been solved and was compared to the crystal structure of the TTR in the absence of the beta 128. It has been confirmed by this comparison of structure that the TTR is able to interact with the beta 128, but we don't know still the exactly sequence of the interaction, but we could observe a change of the EF helix conformation, thus demonstrating the involvement of the EF helix in the interaction between the beta 128 and the transthyretin. So this data, this preliminary data, showed that the EF helix is an interesting sequence for developing new amyloid inhibitors. In our laboratory, we are interested in the design and the synthesis of folded peptidomimetics as previously presented by Professor Ongeri. In particular, we are interested in inserting in peptide sequences non-natural amino acids which are able to induce a particular secondary structure conformation into the peptide sequence. We recently developed a new synthetic scaffold, which is composed by 2 sequential ADSA amino acids followed by 1 natural amino acid. This tripeptide mimetic proved to be able to fold the backbone into consecutive right-handed helical turn. And when inserted in hexapeptide sequences is able to stabilize conformation as 310 helix, which are characterized by consecutive hydrogen bond between residue I and I+3 and by stabilizing 10-member hydrogen bond rings. So we decided to use the synthetic scaffold in order to synthesize new hexapeptide analogs of the EF helix of transthyretin in order to restore the helical conformation of the peptide. We used 1 to 3 D-ADS amino acid motif in different combination in order to study if the position and the number of the D-ADS motif have on the stabilization of the helical conformation of the peptide. And this type of approach brought to a library of at least 10 hexapeptides with the free N-terminal or with the acetylated N-terminal. And all the compounds were subjected to a conformation analysis first by circular dichroism in order to select the most interesting conformation. As we can see here in this slide, compared to other position, the D-ADS motif, when it's inserting the peptide at its in the N-terminal part of a peptide sequence is able to stabilize a conformation that in the circular decreasing analysis show a positive Cotton effect at around 225 nanometer and a stronger negative effect at around 200 nanometer. Comparing this CD spectrum obtained with our peptidomimetics and with the CD spectra that we can see in the literature, we found a similarity of this signature to the 13 helix that are stabilized by foldamers, which contain beta and gamma amino acids. And we observed that this positive Cotton effect that probably is the signature of this type of 13 helix is even more stable and even more intense when two diazomotives are used in the sequence, one in the N-terminal and one in the C-terminal. By assessing the inhibitory activity of our peptidomimetic of all compounds by theoflavine T fluorescence assay, we found that the peptidomimetics that are more stable and they fold in a particular helicoidal conformation, so the compounds that present both the diazomotive at the N and the C termini, were able to delay the aggregation process of the beta 142 peptide, but the activity was less compared to the natural peptide derived by the sequence of the EF helix without any type of secondary structure conformation. Surprisingly, when we removed the acetyl group of the N-terminus, we could observe a complete inhibition of the amyloid aggregation at the same concentration. So at this point, we were facing to a question. If the difference of activity between the acetylated and non-acetylated compound was due to the fact that probably we can establish an ionic interaction with the free N-terminus, or if because we have a different conformation of the helical conformation of both compounds. So in the first attempt, we use again the circular decreasum in order to see if there was a global if in the same condition, both the compounds with and without the acetyl group at the N-terminus could present some difference in the CD signature. And we can see here in this picture that globally in the same condition, both compounds present an overall conformation that is similar, with the same intensity of the positive band at 225 nanometers. So we decided to use the ATR FTIR analysis in order to better understand and to better appreciate a possible difference in the helical conformation of the backbone. And we used the deconvolution of the amide 1 band between 1600 and 1700. And if you can see by comparison of two compounds in water without and with the acetyl group, that the compounds with the free N-terminus is able to present two different types of helix. One that is more the canonical alpha helix structure with the 13 member rings, and the one that is more in a similar way of a free 10 helix structure. So we perform some NMR analysis in water at the 600 megahertz, and we could demonstrate the helical backbone folding of our compound without the acetyl group, as confirmed by some raw intensity ratio of intra-residue and inter-residue alpha proton and amide proton. By making integration of the raw intensity into distances, we could calculate the distances between intra-residue and inter-residue proton, alpha proton and proton amide. And we discovered that the average of the distances is similar to the value of distances that we can find in helical conformation. G coupling constant between alpha proton and its own protonamide suggests a phi dihedral angle around 90 degrees, suggesting in that case, again that a folded conformation is present. While the temperature coefficient were very high, and so no hydrogen bond of protonamide have been found in water, by using another solvent, which is still protic, but still polar, but a protic solvent as DMF, we could see that there are 5 hydrogen bonds that can be established in the peptides, in these peptidomimetics. But if the NMR told us that we had a helical conformation, we couldn't be able to decide which type of helix the 2 diazomotive at this N and C termini are able to establish in the peptide sequence. So we decided to run a dynamic simulation. So the molecular dynamic simulation in explicit water model by using a generalized Amber force field generate a population of 25,000 of conformation, and the first main cluster, that is the first cluster at the lowest energy, represent the 46% of the total population. And this conformant that was generated present 5 hydrogen bonds in the sequence that can stabilize 2 13-member hydrogen bond rings between residue I and I+4, and the 3 consecutive 10 hydrogen bond member 10-member hydrogen bond rings between residue I+3. And the global shape of this peptidomimetics, it was in an helical conformation with a mixture of 13 and 10 helix structure. By comparing the main cluster obtained for the deacetylated compound with the one that was less active but with the acetyl group at the N terminus, we could obtain the first main cluster at the lowest energy that represents the 68% of the total population, thus suggesting a better thus suggesting a more rigid structure in a preference of one conformation. But in that case, it was surprising. We were surprised not to see the correct orientation, the same orientation of the side chain with the cluster 1 of the deacetylated compound, and we observe any type of 13-member hydrogen bond rings in the acetylated analog. And this absent probably induced a different orientation of the side chain. We decided to check the orientation of the side chain in the EF helix of the crystal structure obtained from the analysis of Dr. Ciccone, and we observe that we have an amphiphilic side chain orientation in the EF helix with the 2 lysine that point on one side and the hydrophobic patch composed by a tryptophan and a tyrosine and leucine residue on the other side of the helix. So the fact that we observe a similar orientation for cluster 1 of the deacetylated compound and not the same orientation with the acetylated one, we decide to make overlaying we decide to overlay our cluster obtained from the molecular dynamics with this conformation, with the EF helix conformation in the crystal structure in the presence of a beta 128. And as you can observe, the overlay of the 2 conformation is very good for the compound with the deacetylated, because the RMSD parameters was at 1.4 Armstrong, while the backbone was not completely similar for the compound with the acetylated, with the acetyl group at the N terminus and within RMSD of 2.9 Armstrong. So In this video, you can appreciate that with the similar orientation that we have of the side chain between the conformer one of the deacetylated compound and the side chain of the EF helix with the free hydrophobic side chain that point in the same direction and the polar side chain that point on the opposite direction. And for the acetylated analog, you see that there is no the same orientation, and probably this could explain the different activity of the two compounds against the beta 142 amyloid aggregation. So once we establish the helical conformation of our peptidomimetic analog, we study its interaction with the beta 142 by circular dichroism in order to understand which is the influence that the peptidomimetics has on the conformation on the conformational transition of a beta during its aggregation kinetics. So following the beta 142 conformational transition over time, we can observe that during the aggregation process, we have an increase of the parallel beta sheet amount after 5 hours in condition of phosphate buffer at pH 7.2. When we incubated our peptidomimetic, the beta 124 in the presence of our peptidomimetic, in the presence of the peptide sequence of EF helix and our 13 helix mimetic, we could observe a shift at 210 nanometer, which is correlated by a decrease of the parallel beta sheet amount, which is normally in agreement with the inhibition of the aggregation, because by inhibiting the aggregation, we inhibit the formation of fibers, and so we reduce the parallel beta sheet amount. But by using this so as we can see here, both compounds, the natural peptide and our peptidomimetics, were able to decrease after 5 hours the ratio between parallel and anti-parallel and anti-parallel beta sheet conformation of the full length beta 142. But this circular dichroism analysis of full length beta 142 peptide doesn't allow to explain why we have a better activity of our 13 helix mimetics compared to the parent peptide. So we decided to analyze the conformational transition of the beta 128, because previously we said that we found a very high affinity between TTR and the beta 128 fragment in the presence of copper ion. So as we can see here, the beta 128 during the time doesn't change its global random coil conformation over time. But in the presence of the natural peptide, we have an increase of the helix content at time 0. The same we have at time 0 with our 13 helix mimetics. But what is interesting is that the 13 helix mimetics compared to the natural peptide is able to stabilize this helical conformation even after 3 hours, thus suggesting that probably the complex between our peptidomimetics and the beta is stronger compared to the natural peptide. And this probably is the reason why we have a better activity, inhibition activity. And we decided also to follow the conformational transition of the beta 128 in the presence of copper to see if we could observe any other type of conformational change of the beta 128. So in the presence of copper, our peptidomimetic didn't change its conformational preference. So it was always with this intense positive cotton band and 225 nanometers. So copper doesn't change the conformation of our peptidomimetic. When we follow the conformational change of the beta 128 in the presence of the 13 helix mimetics and the copper ion, we could see that the copper ion alone is already able to increase the beta strand amount of the secondary structure of a beta 128, thus suggesting a negative effect of the metal ions on the amyloid aggregation. But this effect is even more pronounced in the presence of our peptidomimetics. So, but in that case, compared to the beta 128, in the presence of our 13-helix mimetic, we could observe a positive cotton effect at around 240 nanometer that is quite similar to the one that is observed in polypolymeric 2 structure. So we can probably make the hypothesis that probably in the presence of copper, our peptidomimetic might stabilize a mixture of local polypolymeric 2 and extending conformation by trapping the beta, the amyloid peptide, in a transitional conformation that is between an helix and an extended structure. So to conclude, I would like to say that the folded peptidomimetics, as we could see, represent a promising strategies to interfere with the pathological protein-protein interactions, both the beta hairpin mimic and the helix mimetic that we could see in our presentation. And we prove that the cross-interaction design strategies are valuable approaches to obtain potential inhibitor of the protein interaction, both by making inspiration from the interaction from chaperone and amyloid protein, or even from one amyloid protein that has a positive cross-interaction on another amyloid protein. And in general, compared to natural peptide, folded peptidomimetics, thanks to their secondary structure stabilization, we demonstrate that are able to interfere better with amyloid aggregation by preserving the disorder or transiently folded monomer species of amyloid proteins. So for this part, I would like to thank all my collaborators for this project, in particular Chenghui Shi, the PhD that synthesized all the azapeptide. I would like to thank Professor Sandrine Ongeri, Dr. Julia Caffi for the analysis of the data and for the fluorescence assay made by Julia Caffi. And thanks to Professor Tapaduang for the molecular dynamics simulation. And for sure, I have to thank Dr. Lydia Ciccone for the crystal structure of TTR in the presence of a beta 128. And for sure, thank you all for your attention.
Thank you, Sandrine and Nicolo, for the great talks. And the audience, please, you can post your Q&As in the Q&A portion of the Zoom app, and also you can raise your hand so you can be admitted the panelists if you prefer to ask your questions personally. Let's see. I don't see anybody raising. I have a question. I'll start while we are waiting for the questions. I have a question which I was thinking in some of the assays, most of them, you have been showing that the peptides, they don't tend to aggregate and fibrilize. Am I correct? So you're selecting them specifically, all your inhibitors, so they are not self-interacting with one another, correct?
They are not self-aggregating together. You mean the peptides? No, we maybe I can show again some curves, but we check, of course, that our peptides are not able to aggregate, to self-aggregate. Sometimes it can happen, especially with the, maybe if I can share my screen, it happens with the with one compound. Can you see my screen? Yes. With this kind of peptide, peptidomimetics, we had this one. Okay. So with this one, which is very extended, in that case, we had self-aggregation. Okay, but in the other case, no, we check and they do not self-aggregate.
And they are typically speaking not toxic. I know, Sandrine, that you showed at the end of your talk, but Nicolo, what's about yours? They're typically non-toxic.
We need to perform some toxicity test, and we are still waiting for the results of toxicity. Of course, generally we do some cell viability assay to confirm that they are not toxic for cells, and they are not toxic in the presence or even with the beta 142 to see that they rescue the toxicity of beta 142.
And my follow-up question, because this is something you always worry when you work with the peptides. It's really impressive how good inhibitors they are, like, of your work. But have you thought about the brain-blood barrier crossing, and is it in a works? And how big they are? Because I can see that you have quite a bit of range of peptides.
Yes.
And yeah, so let's first start with the brain-blood barrier crossing.
Yes, in fact, we have to check the stability first toward proteolysis. So this is in course for the DL171 that I showed, and it's also in course for the TTR helix mimics of Nicolo and Shenhui. So, and we are also trying to see if they can cross the BBB. In the case of DL171, we didn't see any toxicity, at least on cells. And as we have activity on tau and microtubule, it means that it's able to cross at least the cell membrane. And we don't have toxicity in that case.
And recently, we published a paper about peptidotriazolamide, a peptidomimetic in which you have a triazole ring in some position of the amide bond, and they proved to be able to cross the BBB in a rodent model and even in a human model. And then probably for the TTR, we can say that this fact that we have this amphiphilic helix structure, probably it will help to cross the BBB, because generally amphiphilic helix are able to cross membrane.
Yeah, so thank you. That's I just saw that there is a question in the chat. This is from Bina. Have you tested whether the promising peptidomimetics dissolve aggregates when added to preformed amyloids? That's also quite an important.
So for these two last compounds, no, but for previous one we have tested and they are not able to dissolve aggregates. And this is quite good news, in fact, I think, because probably large aggregates are less toxic. I don't say that they are good. It's not good to have large aggregates in the brain, this is for sure, but they are probably less toxic. And it is reported that maybe by disaggregating large aggregates, you can form more toxic oligomers. So we are not really interested in dissolving large aggregates. In fact, the idea is to try to trap really at the beginning of the misfolding, to try to avoid the presence of oligomers. So it's why we checked by capillary electrophoresis and by IMSMS that we don't have any more toxic oligomers. So at least for the previous that I showed in previous work we had, they were not able. For example, this one was not able, and so this one was not able to dissolve. The previous one neither. So when you have large aggregates, they cannot enter anymore in the beta sheet structure. you have to really interact with the previous folding.
Thank you. All right, there is a continuation, but in such a scenario, how do you propose using such molecules as therapeutic molecules?
Yes, this is a very good question. The idea is really to trust on the early diagnostic, I think. And I think this is the clue and the issue now for anti-aggregative agents in general. When you have very late aggregates, in any case, it's too late. I think the neurons are destroyed in the case of Alzheimer's or Parkinson's disease, or in the case of pancreatic cells. So we try to trust in early diagnostic. I really trust in this. Otherwise, in any case, it's too late when you have very large aggregates. And it is also reported that sometimes some patients, when they have large aggregates and only large aggregates, they don't suffer from Alzheimer's disease. So it's quite a proof that large aggregates are not the most toxic species. So the idea is to try to use it very early. If we have early diagnostic, this is not the case. I agree. But many attrition in the clinical phase come from the late cohorts of patients, I think.
Thank you.
So if we
I just let Bingzu, you can ask your question.
Hi, Dr. Ongeri. I have a question about your compound, DL171. So have you tested its cytotoxicity? It inhibit the tau-induced cytotoxicity. I noticed you mentioned it's not detectable or whatever. And you do mention that it's an Axon transport assay. How does the data look like?
Yes, so we didn't test directly. You mean the toxicity of tau on cells and the inhibition of the toxicity of aggregated tau? No. For now, because in this consortium we are very interested in the effect on microtubules. Okay, so we want to rescue the activity of the physiological activity of tau. So we want to check if we inhibit the aggregation, we can rescue the binding to microtubules. So this experiment is only about that. We did not perform any cellular evaluation of the toxicity of tau directly. Is it?
Yes, thank you. Thank you. I know it's there is not many tau-induced cellular assay out there for testing. No. And I think it's great if you can get showing data showing like it affect or inhibit its toxicity, which can lead to axon transportation, which is also neuronal-based assay. I think there are some study out there, and that would be great also. That's an early indication of like a cellular-based neurodegeneration assay, so related to axon transportation.
Yes. In that case, we just check that we can recover rescue the binding, the physiological binding of tau on microtubules.
Thank you.
So we have a question in Q&A, and that question is from Yangshi. Nice talk. I was wondering how these peptides affect physiological functions of the amyloid proteins, such as interaction between tau and MT. Between tau and? Microtubule.
Microtubule.
And microtubule. So if I understand well, the compound, it's the K-off that I show. So for example, the compound does not bind directly to microtubules. So it preserves the aggregation, and while it preserves the aggregation, it rescues, as I told a few minutes ago, the physiological activity of tau. but it does not bind directly on microtubule. Is it? I'm not sure that I understood really well the question, maybe.
And doesn't interfere the interaction of the tau with the microtubule.
Microtubule, yes. Yes, even in
and
the
this is demonstrated by the K of parameters that it doesn't change.
And the K-on increase. So even if we don't know yet where the compound is bound, we are waiting for NMR studies from Marcus. But even if, now I understood maybe the question, we probably the compound bound to the to the microtubule binding repeat domain. Maybe we will check. We are checking that with Marcus. But it's true that it's a good news, even if it binds to this MTBR, it does not prevent then the binding of tau with microtubule.
Yeah, thank you so much. We have an attendee who wants to ask in person a question, Julia, but for whatever reason, she cannot. Sam, Rams, do you mind helping me? Can you promote her into the panelist?
Somehow maybe I don't know that she's really interested in asking the question or not. I tried to promote her to the panel. It's not working.
Oh, it's not working for you too.
Not accepting it.
Yeah, Julia, please, you can type in your question. We have difficulties promoting it.
Let me ask a question here. Nicolo and Sandrine, great talks. I liked it very much. So when you target the helical intermediates, it seems to be nicely working for A beta and I think one other peptide, I forgot. So what happens to the other intermediates that could also nucleate the aggregation? How does this work?
This we don't know. For now,
this we don't know. Yeah. Because the interaction, the study of the interaction that we perform, it's only with the bulky analysis of the circular dichroism that doesn't give to us an explanation, a demonstration if we have a monomeric or just a small oligomers that they fold in a way that are in an anti-parallel or helix structure.
Maybe
we can see the global conformation.
Yes, but maybe we can hypothesize that, you know, that especially oligomers are dynamic. So they are folding, random coil, helical, and beta sheet structures. So maybe if we trap some helical species, then it forces maybe all the oligomers to adopt and to all the monomers to adopt this kind of helical structures. And we hope that if it happens like that, then it can promote the clearance of this helical monomeric species. But it's just an hypothesis. We don't know. It's an equilibrium between different species, oligomers, monomers, helical, beta sheet, random coil. So maybe when we force the interaction with helical structures, then we force all the amyloid protein to adopt the same helical conformation.
Yeah,
maybe.
Interesting. So most of the oligomers reported in the literature are non-helical. Only there are few non-helical structures, right? So yet your inhibitor is very efficient in interacting with the helical intermediates. As you say, maybe it's converting to form, I don't know, converting all other species to form helical structures. I don't know, that needs to be proven, but it's interesting. I have another question. How do you make these compounds bioavailable? How do you overcome the enzymatic degradation in the in vivo conditions?
We hope that our peptidomimetic were stable, but we will do the test by to see the enzymatic activity, and the proteolysis activity is not able to cleave our peptide. So the stability we will perform by test. But we hope that the peptidomimetics are generally more stable than peptides.
I see. Okay.
And we have hope with at least for the DL171, because as we have activities in cells, we have proteases in cells, and they are active. So probably even if something is cleaved a little bit, you still have activities. For the diaza peptide, it is known, this has been reported, that when you include ASA amino acids in peptide, you dramatically increase the proteolytic stability. So we have hope. I hope I will show you that in Catania. We will show you that in Catania in a few months.
You also showed the selectivity against A-beta, between A-beta 40 and 42, right? Even though your inhibitor is binding to the central region of the peptide, not the terminus, yet it's selective towards A-beta 40 as opposed to 42. How does that work?
It's really because of, I think, because we have two specific sequences. I don't know if I show you the first compounds.
Amino acid
probably because
the same where this inhibitor is binding. The amino acid sequence is identical for the peptides, and according to you, it's binding to the monomers. So
to the monomers, or it really depends. I didn't have time to show you, but by IMSMS, for example, this compound is able to bind also to dimer and to trimer by mass spectroscopy. And so it's hard. It's a hard condition when you do mass spectroscopy, and we can see non-covalent binding of this compound with the dimer and trimer also. But I think that really the selectivity comes from the two sequences, probably, because we have really two different sequences from the IAP in that case. So we do not have any activity on A-beta, not at all. We didn't check on tau and we didn't check on IAP, on synuclein, but now we have all the models, so probably we can check also if we have real selectivity or only selectivity between A-beta and IAP. Between tau and A-beta, I mean, we are not looking for selectivity. If we can avoid the aggregation of both, it's very good, especially because tau and A-beta are cross-interacting and they probably interfere each other, one with each other. So we don't search for selectivity in that case.
Okay, thank you very much.
Thanks.
I have another question for you, because you worked with so many different systems, protein aggregating systems, and then you developed different peptides. Do you find any correlation between the size of your inhibitors with the size of what you want to inhibit, meaning shorter peptides, or like amyloid beta require shorter inhibitors, or there is not such a correlation?
No, for now, we have a minimum of size, I think. We have a minimum of size, but for now, we don't have really correlation. For example, for tau, you see, to inhibit tau, we have more or less the same size that for IAP, while it's much, because I think for tau, not everything is aggregating. It's really the center, only small parts of tau that is folded in beta sheets structure. So probably we don't need very large surface. We don't need to inhibit large surface in the case of tau. So even short, and we are, Nicolo and myself, we are pharmacists, so we try really to avoid big molecules. It's quite big, but we try to keep 1,000 and not more for the molecular weight. We try to keep it's even larger than the Lipinski rules, but we try to keep the same size more or less. But you see in this example, we could also decrease the size and decreased the number of residues, and we had similar activities.
But more or less, we can say that around 10 amino acids length of the peptide are the best inhibitors that we have in hand. More or less, between 8 and 12.
Yeah, thank you. That's yeah, I thought so, but I really wanted to clarify on it. So
thank you for the question.
Rams, do you have any more questions?
No, I don't. I do have, but maybe some other times.
Yeah, and I don't see anybody else besides raising the hand from the audience. Sam, do you have a question? I just want to make sure everybody asks their questions before we close.
If you have questions, you can write to Nicolo and myself.
Yeah.
Easy to find.
Thank you very much, Sandrine, Nicolo. I think we can close the session.
Yeah.
Thanks.
Thank you to you.
Thanks
Thank you very much.
Bye.
Yeah, bye.
Yeah, thank you.
Bye.
Thank you, Sandrine and Nicolo. Nice meeting you.
Thank you, Magda.
Thank you so much. Thank you.
Bye.
Bye.
Nice
See you
meeting you.
soon. Bye-bye.
Bye, P. Bye-bye.